Welcome to Research Goes Viral by the MRC University of Glasgow Centre for Virus Research. My name is Bailey Atkinson, and I am a second-year PhD student based with Liam Brierley and Daniel Stryker. I'm here today with Dr. Ruth Harvey, the Deputy Director of the Worldwide Influenza Centre at the Francis Crick Institute. Welcome, Ruth. How are you feeling today?
Oh, well, thanks for having me. Real pleasure to be here. Thank you.
Absolutely. So before we get started and talking a bit more about your work, I want to give a brief background to our listeners, and feel free to clarify or correct me if anything I say is incorrect. So Ruth Harvey began her career in government laboratories before carrying out academic research in influenza virology and reading at Oxford, and then moving to the National Institute for Biological Standards and Control. In 2019, she joined the Worldwide Influenza Centre at the Francis Crick Institute, where she is now Assistant Director. The WIC, or the Worldwide Influenza Centre, is one of 7 centres in the world responsible for analysing influenza viruses circulating in the human population, overseen by the World Health Organization, or the WHO. And through her work through the WHO, Ruth plays a direct role in deciding which influenza viruses will be included in vaccines each season. She also oversees high containment virology work, including high pathogenic H5N1 viruses. Did that seem like a reasonable overview?
That sounds like a reasonable overview of what I do, yep.
Excellent. Glad to hear that. So just a quick, easy question to start off. Why should we care about influenza virus surveillance? And what insight does it provide for public health or policymakers and the like?
Oh my goodness. Well, I mean, I could talk for hours about flu. Flu is still a really significant disease around the world. It causes over 500,000 deaths every year still. That's just seasonal flu. And the surveillance is so important because we need to understand what's circulating, how the virus is changing, so that we can update vaccines to make sure that they're as relevant as possible. And also the surveillance means that we'll detect those incidences where you get spillover from the avian population. Water birds are the natural host for flu. So where we see those spillovers into the human population, which can cause just single incidences of disease, or could be a threat of a new pandemic virus.
Yeah, very exciting. And I mean, obviously, I'm convinced as a virology researcher, but that's great to give a general overview for the listeners who maybe don't understand how it takes that comes into virus surveillance. To kind of expand on that a little bit, I am wondering if you could walk through me kind of a typical day of what happens when a concerning or maybe even a novel flu strain is detected somewhere in the world, and how you're able to kind of coordinate your collaborations with these 150 plus labs all over the world, and what that kind of looks like.
Well, I mean, yeah, it is a global network. So there are about 150 national influenza centres around the world, and they're going to be likely the labs that are going to pick up a novel case of flu. They'll reach out to WHO directly, and WHO can put them in touch with one of the collaborating centres, which can give them the most help. It can be any of the collaborating centres. It tends to be the one that's geographically closest because it's easier to share materials. If we're alerted to such a case, we will work with the National Influenza Centre in country and give them the support that they need depending on their own capabilities. So obviously the first thing we want to do is we want to characterise that virus. We want to know what it looks like. We want its sequence. And we want to do some antigenic testing. We have a whole raft of viruses in the freezer ready to go as candidate vaccines. And the most important question for us is, would one of those be suitable? If this virus was the virus that was going to cause a pandemic or an outbreak, is one of our ready-made viruses going to be a suitable vaccine? So we'll do that testing. If yes, great, then that we know we're covered. If not, then WHO would consider recommending a new vaccine strain to be made based on the emerging virus that's come out that's antigenically different. Beyond that, it's just whatever it depends on the nature of if it's a single person that was infected from a backyard flock, for example, if it's an outbreak at a farm, or some sort of agricultural setting. It can really depend based on the setting of the outbreak or incident, the level of response and support that the country will need.
Absolutely. Yeah. And you talked a little bit about kind of looking at particular characteristics of that flu strain that might be coming in. What characteristics in particular are you most interested in, and what are you really looking out for?
So, I mean, we're looking at antigenicity. We're really focusing on the vaccine response for that. So we are looking at antigenicity primarily, but also we'll be looking at the characteristics as to, for example, is it a high path virus? You know, the characteristics of it, which we can get a lot of information from the sequence. We'd also be looking at its sensitivity to antiviral drugs. Both by sequencing and by testing, wet lab testing, we can check that the viruses are sensitive to the licensed antiviral drugs that could be available to be used. Beyond that, it would be just understanding its receptor binding specificity. For example, has this virus adapted to be able to spread well in mammals or humans, for example. Right. So there are all of these levels of detail that we'd be interested in.
Absolutely, yeah. And this is obviously very relevant considering the avian influenza outbreak of last year.
Absolutely.
And the dairy cattle. And I think that's pretty much a really common example that I feel like a lot of flu researchers go to now when talking about the characteristics in terms of receptors. Because it's something that we didn't know that beforehand, that there were those receptors in the mammary glands.
Absolutely. I mean, flu never ceases to surprise you. We think we know everything about how it's going to behave, and then all of a sudden it's infecting cows for the first time. And this was a whole new experience for us, and understanding how it was infecting them, infecting the udders, why it wasn't then spreading, and all of those characteristics of that outbreak, and the ongoing risk that it has of spillover to poultry or to humans.
Absolutely. And this is a bit of a broad question, but in your external seminar, you had a slide that showed essentially, generally, how widespread flu can spread among different animals, and it can spread between different animal species. Are there any animals that you may be thinking could be the next potential host, or is that too close to tell?
I don't know. I mean, we've had dairy cattle. Yep. We had an infected sheep in the UK last year, also in the mammary tissue. So, I mean, it could be others. You could be looking at any animals like that in an agricultural setting, goats, other milking animals. Oh, goodness. Marine mammals are often a risk. They do get flu. We've had flu isolated from whales and seals in the past because they have that interface with the wild water birds. So it could be anywhere, and I don't think I'd like to guess the next species.
Fair enough, yeah. I mean, it is also something where, it can come out from any direction, it seems.
Absolutely could, yeah.
So kind of going back a bit more to surveillance. We often hear a lot about surveillance success stories, and I'm kind of wondering if there are any gaps in surveillance, if any.
I mean, I think that there's always more you can do. I think for us, the really important interface is that animal-human interface. And a lot of those interfaces happen in countries that maybe don't have such good funding for surveillance. So, I mean, we work, our director, Nicola Lewis, comes very much from the veterinary side, and she has a really personal interest in a One Health approach. And we do work with colleagues from the animal sector increasingly to really strengthen that. So I don't know that it's a gap, but I think it could be strengthened. And we are actually working to do that, to make sure that we're really, really getting a better overview of everything that we can see in animals, especially where they are coming up against humans in whatever setting that is, and making sure that we fund that in countries where it's really super relevant.
Absolutely. Yes, that's super interesting. And I think I'm also biased because I'm also interested in looking at the human-animal interface when it comes to virus transmission. But I think that's critical. Absolutely.
Absolutely. Yeah.
Clearly. So kind of going into a bit more about your career and your career path. I'm wondering about if there was any particular moment or experience that you had that kind of ignited your interest in working with viruses in general.
Well, I actually can pinpoint where I realised that there was this whole world of science for public health that wasn't necessarily being a medic. And I was actually really, really lucky. When I was at school, I did work experience, and I was sent to a hospital lab for 2 weeks to do work experience. And all of a sudden, there was this world of lab work and science that was public health related. And I always had an interest in infectious disease, but you didn't have to go and be a medic and do all of that, medical stuff. There was a whole world of being in a lab, hidden away, doing really cool stuff for public health. So I think that's the key moment for me. And it kind of went from there. I think I fell into flu because Wendy Berkeley was good enough to give me a job in her lab. And from then on, it's such an interesting virus and there's always so much to learn and it still surprises us.
Yeah.
So some years later, I'm still here.
Absolutely. I've always told this to my friends, like if I were to take a different career than what I'm doing right now, it probably would be like an infectious disease doctor. So I totally relate to you. When it comes to that, I feel like growing up, I had that expectation that if you're interested in science and you want to work in science, being a doctor or a physician is kind of the big career that gets pushed to you a lot.
Absolutely.
But there's so much more out there.
So much more that you can do. Kind of behind the scenes, maybe, which is a very comfortable place to be.
Absolutely. And so going along to your career in terms of the skills that you were learning throughout your career path, I'm wondering which skills or experiences that you thought were the most valuable to get to where you are now.
That's a really hard question. I mean, I think for me, it's being methodical. I think a lot of my early background was in assay development. So you had a really methodical approach to each aspect of that assay to get it working. And I think the same approach works for infectious disease. If you've got a new thing, when COVID came along, we had a new virus and it's working out, okay, it's going to grow in some kind of cells, for example. So what cell type is it going to grow in? And it's that really methodical approach and breaking massive questions down into single distinct questions that you can answer quite simply and methodically. And I think that's always been the approach that's worked for me. And I am still very hands-on and very happy in the lab. So just jump into the lab and try something and see what works and keep working until it's done.
Yeah, that's some great advice. Yeah. I think working methodically is something that admittedly I need to work on a bit more.
It sounds quite dull. I mean, I love a list. Anyone who knows me will know I love a list. But that, for me, it works for me. I haven't got that sort of really big picture approach necessarily. It's that breaking things down and really, having that approach to work through a problem. But it certainly works in the applied field.
Absolutely, yeah. And it can also be applied to other aspects of life too, like baking. It's such a methodical approach.
It really is. Yeah. You've just got to tweak one ingredient at a time and see what happens.
Absolutely.
Yeah.
Well, thank you so much for your time. I just have a bit of a fun question to end the podcast. I was wondering if you have any plans to explore Scotland or in Glasgow while you're here.
You know what? I'm on a train back to London this evening, but I did get to go for a nice walk around the Botanical Garden this morning.
Oh, lovely.
It was really nice. At least I got to see something.
Yes, and you were able to be warned.
Exactly. Yeah, but no, it's straight back to central London, sadly, this evening.
That's unfortunate, but maybe that's an excuse.
Next time. You'll just have to invite me back.
Absolutely. Yes, we would love to have you back. And thank you again so much, Ruth, for joining us today on the podcast.
Time. Thank you.
Absolutely. And thank you, listeners, for tuning in to this week's podcast. And we hope to see you around.